Previously, we talked about the protein structures and we talked about the bonds that do maintain the protein structure, the unique, the van der Waals forces of interaction, and then the hydrophobic interactions, and then the hydrogen bonds. Then we illustrated the bonds right from the previous video or the previous lesson, and the video is there. You guys, you can go there and watch it and enjoy. Then we talked about the structures of proteins, where we talked about the primary structure, which is just a sequence of amino acids that are held together by a peptide bond. Okay? Then we talked about the secondary structure, where there is some coiling and you coil to form the alpha helix and the beta pleated sheets. Okay? And some she bonds are involved in that. Then the tertiary structure, where you further coil and form that 3D shape. Then the quaternary, you have more than one chains to form your protein. Then we talked about types, where we talked about the fibrous, the globular, and then the properties of each. Then intermediate proteins, which are both fibrous and they are soluble and insoluble. Then we talked about the conjugated and we gave examples. So basically that was our previous lesson. So this is where our today's lesson is going to begin from. Okay? So welcome to another lesson. This is our today's lesson. The continuation of proteins. Okay? And let's classify our proteins based on functions. Okay? And like we said, these proteins do actually perform different functions in our bodies. Okay? And so based on those different functions, then we can classify our proteins. So number one, we can classify proteins as enzymes. Okay? As enzymes. And enzymes, these are biological catalysts that do speed up the rate of a chemical reaction in our bodies, both plants and animals. Okay? And we can classify them. We are going to study enzymes as enzymes.
Excuse me.
Yes.
Are there enzymes also in plants?
Why not? So you mean plants
I've
don't have
heard,
enzymes?
I've heard it for the first time.
Oh my god. So you mean plants don't have physiology? Physiological reactions? Or metabolic reactions? Do they have metabolic reactions, plants?
They have.
Such as, number one? Okay, such as photosynthesis. Okay? Such as respiration. All of these are metabolic reactions. Okay? Such as amino acid synthesis, protein metabolism, very many, lipid metabolism, very many. Synthesis of chlorophyll and stuffs like that. All those metabolic reactions in plants, they are enzyme controlled. Okay? So plants too have enzymes. So if you've had it now, it's good. Now you know that plants do have enzymes. And we shall have topic enzymes. Okay? Actually, I think today we shall talk about enzymes. And we shall really appreciate what enzymes are and their biological importance to our lives. Then proteins can be structural. Okay? Yeah, they can be structural. For example, those that form the hair, those that form the hooves, the scales, the fingernails, and stuff like that. All those are structural proteins. The muscle fibers, all those are structural proteins. Okay? They form structures. Okay? Yes. So they can be categorized as structural proteins. Then they can be, we have signal proteins, the hormones. Okay? And these hormones, what they do is they do transmit, they do transport signal or a given message from one point to another point. Okay? And these hormones are so important in our bodies. And we shall study the endocrine system, by the way, in coordination. But we have really made videos.
Excuse me,
Yes.
you had breached. I didn't hear where you started the signal protein.
Oh. So I said the signal proteins, they are hormones. They do transmit a chemical message from one point to another point. Okay? From one point to another point. And this message, it's really important, okay? A moment, this guy is. So they do carry a chemical message from one point or from one organ to another organ. Okay? And hormones are so important as far as homeostasis is concerned. Keeping our internal environment constant is concerned. So they really do play an important part. And we shall study hormones as far as coordination is concerned, but we have made videos, all videos are in our classroom. So you can go there and watch coordination and become an expert even before we study coordination. Then these proteins, we have contractile proteins, the proteins involved, okay, in movement, such as the myosin and the actin fibers. They are involved in movement as far as contraction of muscles, relaxation of these muscles is concerned, and they bring about locomotion. You are able to locomote from one region to another region because of the muscles. And these muscles you have there, these structural proteins, the myosin and the actin filaments, whose contraction and relaxation bring about shortening and lengthening of the muscle, which brings about the overall contraction of the muscle unit, which brings about either locomotion, folding of the arm, able to speak, moving up and down of the jaw, and stuff like that. All those, it's proteins. Then we have storage proteins, the albumin in eggs. Okay? Yes, you know that we don't store proteins, and no animal can store proteins. Okay? Yes. But you know, as a growing chick, the egg must have enough nutrients to support the growing chick to grow within the egg until when it gains a given size to come out. So now, therefore, you must find ways, you must devise means of storing these proteins. So these eggs, or the proteins in eggs, are stored in form of albumin, and it is a protein. Okay? Yes, that's why when you boil an egg, it solidifies because it is a protein. Okay? And we shall see why you heat an egg and it solidifies. But you heat other things, maybe ice, it melts and turns into a liquid. Okay? Then we have defensive proteins, such as antibodies. Okay? Now antibodies, they are proteins, real proteins. and they do defend your body against external pathogens. So this is really good. That's why, imagine if you don't have antibodies, really, if your body cannot produce antibodies, then I don't know how life would be. Okay? I don't really know how life would be. So these antibodies, they really defend our body. That's why you go for immunization. You do immunize. For instance, coronavirus, how they are encouraging everyone to immunize those who are entitled to it. But they have you immunized or you are not entitled?
I haven't.
So why do people immunize? Why do you immunize? To develop antibodies so that on the real exposure you are able to respond. So you are able to respond, okay, when the real pathogen comes in. And so those are the antibodies of ours. Then we have transport proteins, e.g. hemoglobin and myoglobin, but specifically hemoglobin and other respiratory pigments. These ones, they are proteins. You remember when we tried to talk about hemoglobin? Said it is made up of 4 polypeptide chains. You see, 4 polypeptide chains. So it is a protein. And you know how hemoglobin is important to our bodies. Okay? So wake up one day and you don't have hemoglobin and see how you will supply your body with enough oxygen. Okay? Yes. So roles of proteins, we can get them from what we have just discussed. They can be structural and metabolic functions, whereby if I talk about structure, we have the fibrous proteins that do perform structural roles. They are tough, made up of parallel strands, okay, insoluble in water, hence adapted to perform their structural roles. Whereas the globular or the metabolic roles, we have these enzymes, the hemoglobin, all globular proteins, and all globular proteins are soluble in water to form that colloid solution. Why are they soluble? Because they do, remember, their function is, it's metabolic, okay? They do coordinate these metabolic activities in our bodies. So you must be able to dissolve in the aqueous medium so that you are able to catalyze the reaction. So all these globular proteins, they are highly soluble and they are highly responsible for maintaining their metabolic activities in our body. So for instance, we have enzymes that have that globular structure with a specific shape, okay, for catalysis. We are going to talk about enzymes in the few slides. Then we have hormones. For instance, these ones, they are also globular, soluble in water, and they do regulate the metabolic activities. They do maintain the homeostasis of our bodies, maintaining the internal environment constant. So there are these hormones that do play an important part. Then we have respiratory pigments such as hemoglobin, myoglobin, and other respiratory pigments. These ones, they are globular, soluble, and they do have a quaternary structure. Remember that quaternary structure, where you have more than one polypeptide chains within a protein structure. Then we have immunoglobulins. These ones, they do bring about the immunological responses to be able to respond to a given pathogen. Okay? Then we have the myosin and actin filaments, which are responsible for muscular movements. muscular contraction and relaxation to bring about movement. Then we have the globular proteins which do buffer, okay? And remember when we talked about the buffer and how the proteins are able to buffer, and buffering means maintaining the internal or the pH of a given solution or solute or solution constant or fluid constant when a little acid or little base is added. So you resist changes in pH, and these globular proteins, they really do that so much. For example, hemoglobin, it will exist as a sweeter ion that at a certain point you have both sorry, the ionized acid and the ionized base, so that if a little base is added to bring the OH ions which would alter the pH, then the OH are buffered. Similarly, if the little hydrogen ions that would alter the pH, okay, are added, then the hydrogen ions are buffered. And so you maintain a stable, constant pH for these chemical reactions, for these metabolic reactions to take place in our bodies. Okay? Yes. Then transport, the lipoproteins. They are responsible in lipid transport or fatty acid transport, whereby you have a protein, okay, within blood, within serum, that will combine these triglycerides to form a lipoprotein, so that you can easily transport these lipoproteins throughout the entire body. Otherwise, you know that fats, they are insoluble in water, and we know that almost, is it 80 or 90? Fact check that. 80% or 90% of blood is just water. I think it is 70. You can fact check that. 70% of blood is water. So therefore, since fats are insoluble in water, then it will be hard to transport these fats in water, in an aqueous medium. So what do you do then? Then you combine your triglyceride with a protein to form a lipoprotein, which the lipoprotein now is soluble in the aqueous medium and can easily be transported in blood. So as you are transporting that lipoprotein, then you are transporting also the lipid. Okay? Yes. So basically those are the roles of proteins in biological systems. In case I set such a question, then guys, you should really write an essay that is worth 20 marks. Okay? Yeah, let's talk about protein denaturation. Hmm? I think in O level you are told that when you are explaining how heat or how temperature affects the rate of enzyme activity, you talked about an optimum temperature, and then you agreed that beyond the optimum temperature, then the enzymes are denatured. But were you told what actually denaturing is? Denature. Hmm? Proteins are not killed, but they are denatured. Okay? And so denaturation is simply the phenomenon by which a given protein loses its 3D structure. As simple as that. If you have your protein and a protein has such a structure and a structure has such a shape, and this is its active site, okay, then if certain forces happen, or if something happens, and you make this enzyme or this protein, not only enzymes, but this protein, okay, to lose its shape, okay, even if it is some slight change, and you lose the shape of this active site, then the protein is going to be considered denatured. Why? Because it is rendered useless. It cannot carry out any metabolic, any catalytic activity, because this is its shape. And so the active site, the substrate for this enzyme must have a complementary shape. Now someone will say must have a same shape. That one is wrong. It must have a complementary shape so that it successfully bind to this active site. So if something happens and you change the shape of this active site, then this enzyme will be rendered inactive, okay? It will be rendered denatured, and so it cannot carry out any catalytic activity. So this phenomenon whereby a protein loses its 3D shape is what we call denaturation. Then, in some cases, the protein can remodel again and form back the shape and form back the protein, the other protein. Now that process is called renaturation or renaturation. Okay, that is when a protein remodels back and forms its own shape, but it depends on the degree of denaturation, and it also depends on the factor that caused denaturation occur. Okay, not all the time that when you denature a protein it will regain its shape back. No, not all the time. That's why you boil an egg, and if you cool it down, it cannot become an egg again, but it remains a solid. So that is denaturation, causing a protein to lose its 3D shape. So think about how these proteins, they do have the bonds, like how we talked about the structure of proteins. They do have bonds, okay, that do maintain the 3D shape of a protein. Okay? Now, if something happens and these bonds are broken down, then obviously the protein will lose its 3D shape. Okay? If something happens and these bonds are broken down, then the protein is going to lose its 3D shape. And the protein, these bonds being weak, they are not as strong as, I don't know. So these proteins, these bonds being weak, any force, any agent, okay, that may cause bonds breaking down, it becomes an agent of denaturation. Okay, and so when they are broken down and you lose the 3D shape, then the protein is said to be denaturized. Not so? And denaturation can be permanent or it can be just temporary. It can be permanent or it can be, you can break and lose the 3D shape, but later you regain the shape. So it depends on the factor that is causing the denaturation to occur and the degree of denaturation. So let's briefly talk about factors. And I'm sure I can say, okay, let's not take things obvious. That is what teaching. That is the principle of teaching. I was going to say that you know some, but when you are teaching, you assume that students don't know. Okay? So literally, you don't know. You don't know. So the first factor is heat. And that's why you maintain your body temperature constant. That's why you are a homoiotherm. This is I. You maintain your body temperature constant. Why? Because you do have these enzymes, okay, that maintain the physiology in your body. So anything that will affect these enzymes, it affects you. So one factor, heat. So heat also affects these proteins. Heat affects these proteins so much. One, these proteins need heat. For instance, if it's enzymes, for example, if it is antibodies, for example, they need heat. That's why temperature is maintained high. That's why when you're sick, the temperatures become high. Eh? The temperatures are raised from 37 to around 40 there. That high temperature favors these antibodies to combat the pathogens out of your body. The heat, the enzymes. If temperature is low, the rate is very low, depending on how low the temperature is. But as temperature increases up to a certain point, then the rate increases to maximum. And that temperature at which the rate is high is known as the optimum temperature. But beyond the optimum, my dear, then the reaction decreases rapidly to zero. Why? Because our heat is going to denature a protein. It is going to denature proteins that are catalyzing that reaction. Okay? And if the protein is denatured, then you cannot see any reaction. So now the question is, what's up with heat? What does heat do? Okay? What does heat do that lead to denaturation? Now it's simple. When you have, remember your proteins is made up of amino acids, and amino acids made up of different atoms, carbon, okay, different atoms. You have the R group with atoms and hydrogen with atoms. So different atoms. Now when you subject this protein to heat, now heat is a form of energy, okay, and from the law of conservation of energy, you cannot create heat, neither can you destroy heat, so it can be converted from one form to another. So the same thing, when you subject heat energy to a protein, then the atoms in the protein, they are going to absorb the heat energy, okay, and if they absorb the heat energy, then their kinetic energies, the kinetic energies of these atoms is going to increase, such that these atoms, yes, it's a solid, you know, atoms in a solid are closely packed together, so they don't move past one another, but they can vibrate within their main positions after absorbing energy. So similarly, even atoms within these proteins, this protein, after absorbing the heat energy, the kinetic energy is going to increase, so that the atoms will vibrate they will increase the vibrations of these atoms within their main position. And remember, the overall structure of a protein is maintained by the bonds, whereby you might find here you have a sulfur bridge, here you have maybe a hydrogen bond, here you have the van der Waals forces of attraction, and here you have the ionic bond. So remember, you have the bonds, and the bonds, they are the ones that do maintain that 3D structure, that 3D shape of an enzyme. So if you increase these vibrations within the atoms, then you will reduce the strength of these bonds that are formed between the atoms. And if you reduce the strength of these bonds, that if the vibrations increase, then the bonds will end up get broken. And if the bonds are broken, then the structure which was this with such a shape, it will lose its shape, okay, and become something. And such a structure that has lost its shape, it's rendered useless. Okay, it is useless as far as physiology is concerned. It cannot carry out any physiological activity. So it's as simple as that. So that's why you are a homoeotherm. That's why you are a warm-blooded animal. Okay? You don't require too much heat that would break your bones, that would denature your proteins. Okay? And if you denature the proteins, then you lose their 3D shape and they are rendered useless. And you can also try this at home. You can try this at home. When you boil an egg, it solidifies. Can it solidify before boiling it? No, it can't. Normal egg, it's a colloid. It's colloidal, it's a colloid solution. But once you put it on a pan and you boil it, then it will solidify. Why? It's because you are denaturing the albumin. You are denaturing the protein in the egg. Okay? Now when you denature the protein in an egg, now the protein, the protein within an egg, it is soluble. The albumin, it's a globular protein. Okay? And it is soluble. Okay? But when it is denatured, it loses its 3D shape and forms something. And this something is insoluble. Okay? This something is insoluble. So that's why you boil an egg and it solidifies. Even when you mix it with water. You boil, the egg solidifies, and the water remains on the saucepan. Because the heat is too much and the protein is denatured into an insoluble form of a protein. So it's as simple as that. Okay? It's as simple as that. Then let's talk about some other factor, and this is acid. And with this acid, before we even talk about acids, let me first just use this. I think, is it yesterday or yesterday but one? I have this guy of mine, this friend of mine. He was like, during the corona period, they told him, they know, actually all of us were told that we should eat, we should drink ginger, we should eat lemon, we should eat fruits. You can imagine. And stuff like that. Garlic. Some people would even put red pepper in tea. You can imagine. Now this friend of mine, so because of this, he bought his milk. Okay? He bought his milk And then the milk, he put ginger, he put stuffs like that. But surprisingly, or not even surprisingly, fun enough, this friend of mine also added lemon in his milk. But the worst part of it is that the moment he added the lemon in the milk solidified. And the guy just poured his milk. He didn't take the milk. It just turned into solids. It solidified. Can you imagine? And the guy poured his milk. And when he just made that, I was like, I must use that as a teaching aid. And it's really a nice teaching aid. So you can also try it at home. Buy your milk and then you add their lemon and see what will happen. So why did the milk solidify? We call it souring of milk. Why did it solidify? It's because this lemon is typically acidic. Look for the filter paper, not filter paper, the litmus paper, and test for its pH. You'll be worried. That is really acidic. Around 4. You can look for the pH, the indicator. The other one which is in liquid form, not the litmus paper. The indicator solution, I think. And test for the pH of your lemon. You'll be astonished to find that the pH is around 4. And that is too much of an acid. So when you add the acidic, the lemon, which is acidic, to the milk, the acid will produce hydrogen ion, and hydrogen ions will cause denaturation of the protein. So these acids, they do denature proteins. Okay? And the same as bases, they do the same. They also denature our protein. And so it is just simple. You know that protein, you have their ionic bonds, okay, which are formed between the COO and then the amino group, okay, which has received a proton. So when you add there an acid, the hydrogen ions from an acid, they will react with the COO to form an dissociated acid, which will break the ionic bond. And when you break the ionic bond, then the protein is going to be denatured. As simple as that. And when the protein is denatured, then it will coagulate. And that's how even when you leave milk, that's how it goes bad. These microbes, they will reduce the pH, okay, of milk, which will end up coagulating. They are going to live in the milk, respire, produce carbon dioxide. Carbon dioxide decreases the pH, and then the milk coagulates. So that's why you should maintain your milk safe. Then we have bases. I think that's what is next. Yes, alkali. And this alkali will produce the OH group, the OH ions, which will react with amino group to form undissociated or unreacted ammonia, which also interrupt with the ionic bonds. Okay? And ionic bonds, what will happen? The milk will sour. Okay? The milk will sour. And the protein is denatured and the milk sours. So, guys, this is something simple. Then we have inorganic chemicals such as heavy metals, e.g. mercury, sulfur, and all heavy metals. They combine with these groups, okay, to destruct the ionic bond that is formed. That's why these groups are used as poisons. Hmm? You know, we have poisons. These rat poisons, most. However, we no longer use these heavy metals to form poisons. But these heavy metals can be used as poisons, whereby when you take it, then it will combine with your enzymes in the body, and the enzyme will lose its 3D shape. And if the enzyme loses its 3D shape, then man, the enzyme will be denatured. And if the enzyme is denatured, then it won't carry out the physiology and the organism dies. For instance, in respiration, we have there an enzyme called acetylcholine esterase. Okay, not respiration, sorry. It is acetyl-CoA. First leave acetylcholine esterase. So now these heavy metals, for example, mercury, also cyanide can do the same. I think it's what is next. Yes, also cyanide can do the same. So they combine with that enzyme acetyl-CoA, okay, which prevent it from combining with an acetyl group. And so you cut off the Krebs cycle, you cut off the electron transport system. Hence you just depend on glycolysis, which produces just little energy that cannot sustain you and you die. Okay? You die. Yes. So there is a video as far as respiration is concerned. So you can go and watch it and appreciate that acetyl-CoA. Then organic solvents, they do alter the hydrogen bonds. For instance, if you add there alcohol, the alcohol will form hydrogen bonds with the neighboring hydrogen bonds of the protein, which will disrupt the hydrogen bonds that are formed within the protein. And so the enzyme or the protein will be denatured. And that's why alcohols are used in sterilization. However, not as sanitizer. Anyway, it's the same thing. Now, as sanitizer, you know the alcohol, the membrane, the virus, the coronavirus is just, all viruses in fact, it's just a membrane, okay, cell membrane with some capsids, okay, protein cap with protein spike proteins, okay, not capsid. Some of them are enveloped, others are not, and then a nucleic acid. So what alcohol does, alcohol is CH3, okay, and then CH2OH. So the alcohol, it will dissolve this membrane, okay, whereby the polar groups, the membrane is a phospholipid, it's made up of phospholipid heads. So the polar groups of the phospholipid, they will interact with the OH, the polar group on the alcohol, and the non-polar groups on, I think, okay, yeah, the non-polar groups, they will react with the CH, this carbon chain, and you end up disrupting the membrane. And it's the same case how soap works. Okay? Soap is a long chain of carboxylic acid. Okay? Soap is sodium ethanol. But the chain, this hydrocarbon chain is very long. So it also works the same way. Whereby the polar head, they will dissolve in this polar group, and then the non-polar of the membrane, they will dissolve in this hydrocarbon chain. So that when you wash away, then the membrane is completely disrupted. Okay? And you survive the virus. That's why washing was really good. But also alcohols, the alcohols will also interact with the hydrogen bonds, okay, within this protein, and you end up denaturing the protein. Then finally we have the mechanical force, and this is what ladies use so much. Even you, if you know you grow hair, I'm sure you have a lift at home. Okay? You get the lifter and lift your hair. So as you are doing that, you are mechanically breaking the hydrogen bonds within the hair. You are breaking the bonds, not only hydrogen bonds, but all bonds that might be formed. Okay? Within the hair. Remember here it's a keratin. It's keratin, which is a structural protein. Okay? And this keratin, so as you are lifting, you are breaking the bonds. But now this one is temporary. That's why when you don't lift again, then the hair goes back. Then you lift, the hair becomes good. Then you don't lift, the hair goes back. So that if you spend a week without lifting, the hair will be completely coiled. Can you imagine? Highly coiled. Why? Because the bonds they would have been formed, which will strongly bind the protein close together. So this similarly, ladies go to dryers. There they do apply heat, but also some mechanical force to break the bone. Okay? So that the hair straightens. Okay? Yes. But why do you do that? To break the bonds that hold the hair molecules, the keratin, the alpha helixes, the helixes in the carbon, in the keratin molecule, so that when you break them, then they straighten. And when they straighten, then that's when you see a lady feel proud that I have some good hair, forgetting that she has broken the bones. Naturally, it is not supposed to be that. Okay? Yes, it's not supposed to be that. Others apply jelly, is jelly or whatever. So there, I think there is a chemical. Those jellies or those they contain certain chemicals. Those gears they contain certain chemicals that break these bonds. That's why you have to add jelly. You have to add this gear every day, every day, every day. Otherwise, if you don't add, then the bonds will reform again, and you have no more hair like ours. So basically, that's denaturation, and that's how simple it is. Then with renaturation, here you are forming back the 3D structure of a protein after denaturation. However, like I told you, the degree of denaturation matters, and the factor that has been involved in denaturing that protein also matters a lot. The factor and the degree. Okay? Whereby if the degree of denaturation is not that much, then the bonds can form again, and you have the 3D structure, or you have your shape. For example, like I told you, when you lift your hair, and if you don't lift it for the whole day, and then the whole night, tomorrow it will be highly coiled again. So that is renaturation. So similarly, these globular proteins also, when the degree of denaturation is not that much, okay, then it will regain its shape. But however, if it is greatly denatured, then trust me, you cannot regain the shape. Not so? Yes. So you have some assignment here. Please take a screenshot. You can do. I want you to discuss the role of proteins in nature. And in nature, I gave you I gave you some roles. But I want you to discuss the role of proteins. in nature. Okay? How these proteins are involved in nature. And talk about nutrition, and for instance, you talk about all proteins involved in nutrition. If there are enzymes, talk about enzymes. If any other protein, write it. And then after that, you talk about the function, what that protein do in nutrition. Then you go to respiration, you talk about all proteins involved in respiration and transport. All proteins, if there are enzymes, write enzymes, and you can also write some examples of enzymes that you know or that you come across, and the functions of all those enzymes. If they are hormones, do the same. If they are antibodies, do the same. If they are pigments, do the same. Then you go to excretion, you go to growth, you go to locomotion and movement, you go to sensitivity and coordination, reproduction, and others. Okay? In both plants, since you say that you didn't know that plants contain hormones, then you also go to plants and demystify. Now, when you talk about a given process, a given life process such as nutrition, look at how nutrition occurs in animals, how nutrition occurs in plants, in fungi, and stuff like that. Okay? And cite out the roles of proteins in each. Otherwise, if you can do that, then it will be some good assignment, some good work. Now, in our next lesson, we shall begin from enzymes. Okay? We shall talk about enzymes.